lab test. Okay, and we're going to be flipping back and forth between the handouts and the PowerPoint. So this is really talking about the answer to the essay question. What happens when the action potential arrives at the muscle cell? So we are going to give you the answer to the essay question here in just a second, as soon as the big screen pops on. There we go. So the first thing we're going to look at is the sliding filament model. So what is that exactly? Well, when we're talking about the sliding filament model, we're referring to the fact that the actin and myosin don't change length, they slide over the top of each other. That's the sliding filament model. The sarcomeres shorten, but the actin and myosin do not. They don't change length. So I want to show you a kind of an animation so you can kind of see that happening. And then, of course, with muscle relaxation, the sarcomeres are going to do the opposite. They're going to lengthen, and that's usually by an opposing muscle that's pulling rather than, or extending rather than flexing. So we have this animation here that I want you to take a look at. We're not actually going to watch the whole animation here. We are... It's not wanting to click on it. There we go. We're just going to watch part of it. So you're not going to be able to hear anything. I'm not turning the sound on. Catch it there. So what we're looking at is, so again, I'm not showing you the sound. You have the green, which is representing the actin. You have the purple, which is representing the myosin. You can see two sarcomeres in this image. You remember the sarcomere goes from Z line to Z line, so that's one sarcomere. And then you got the Z line going to the other Z line, that's the other sarcomere. So you're seeing two sarcomeres in the picture. I'm not worried about all the other labeling. So what I want you to do first is just look at the green actin molecule while the muscle is contracting, and I want you to ask yourself, is the actin getting shorter? So that's really all we're looking at. Then we're going to look at the myosin. We're going to watch this 3 times. Then we're going to look at the myosin. Then we're going to look at the thing as a whole, because it's got so much going on, so much moving, it's kind of hard to figure out what you're seeing. So let me go ahead and let it play so you can watch the green actin and ask yourself, is it getting shorter? So the actin, the green, it didn't get shorter. I mean, they came closer together, right? With actin, the end of one actin came closer to the end of the other actin, so they came closer to each other, but nothing got shorter. So now let's back up and take a look at the myosin now, which is the purple. Now, keep in mind the myosin heads are going to be flexing and extending, so you got movement there. But if you look at just the rod portion and ask yourself again, is the myosin getting shorter? So look at the purple this time. You can see the myosin heads flexing. But the myosin itself did not get shorter. So the actin and myosin don't change length. They have what's called the sliding filament model, where the actin and myosin slide over the top of each other. So if you look at the overlap in a relaxed muscle between the actin and myosin, and then how much non-overlap there is, watch what happens as the muscle shortens at the amount of overlap there is between the actin and the myosin molecule. So you can see they're overlapping more as the myosin is pulling the actin over the top of it. You can see there's more overlap, which is actually causing the muscle to contract. So when we come back over here and say let me click the button there. The sarcomere is the smallest contractile unit of a muscle cell. It is what actually does the contracting. This should be familiar. This is kind of a review on Chapter 12, so you should recognize the spinal cord here. All this is saying is the muscle cell has electrical properties, meaning that muscle cell has the ability to receive that action potential. the axons of those neurons are going from the brain or spinal cord to the skeletal muscle fiber. Remember, this right here is that motor neuron, which is communicating with the effectors. That's all that's saying. It's going from the spinal cord here, going out to the effectors. There's the head of that, or the cell body of that motor neuron. We're going to take a look at this animation because you still have to know this for this test. You have a handout. It is this one right here, and we're just going to kind of label the parts. You should recognize this. This is what you responded to your essay question for chapter 12. So this is review. We have our, just to kind of label everything so you see what's going on, this is the neuromuscular junction here. You can see this cell right here. This is the axon terminal of the motor neuron. And remember, we call that the pre-synaptic cell because it comes before the synapse. So this area right in here is the synapse. So that's the presynaptic cell. We have our synapse. And then this area right here all this is the muscle cell or muscle fiber. And remember, we call that the post-synaptic cell, right? It's the cell that's receiving the signal. So this is kind of our bearings here. This is what we're looking at. Now the action potential, this should sound really familiar. The action potential is coming down the axon terminal of the motor neuron. When it hits this protein right here, this is the voltage gated calcium ion channel. Remember that. So when the action potential arrives, calcium will go into the cell. These right here are vesicles, and they are filled with neurotransmitter, acetylcholine in this case. So when the calcium shows up, those vesicles filled with acetylcholine are going to undergo exocytosis and dump the acetylcholine into the synapse. Again, should sound really familiar. The acetylcholine is going to cross the synapse and it's going to bind to this protein right here, which is the ligand gated sodium ion channel. And it's going to allow sodium ions into the muscle cell. That's going to depolarize the muscle cell, reestablishing the action potential. Again, sound familiar? It should. This was the answer to your essay question on the previous test. Okay, so that's just review. We're just labeling the diagram there, so that way you can remember. You still have to know this material in the form of, multiple choice questions. So let me show you the video. You should be able to actually be the narrator of this video at this point.
Potential attacks at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of late-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
Okay, so all of that should be familiar. The only thing different in this scenario than what we learned in chapter 12 is the receiving cell, this postsynaptic cell here, is a muscle cell instead of a neuron. That's the only difference. Everything else is identically the same. So that is the muscle cell receiving the action potential. Here's another video that shows the identical same thing, just had some different animation in there. These are the steps, in case you needed to remember the steps of what happens when that action potential arrives at the muscle cell. Now, we're going to take a look at this handout. Which is action potential and muscle contraction handout. Now that the muscle cell has received the action potential, what happens next? So let's just kind of get our bearings here on what we're looking at. This right here is the muscle cell or the muscle fiber. You've got the plasma membrane of the muscle cell. Now, the fancy name for plasma membrane is sarcolemma. Anytime you see that prefix sarco, you know it's got something to do with a muscle cell. So this action potential is going to go down the plasma membrane of the muscle cell. But remember, we want that action potential to not just travel along the surface, but to be able to get in deep into that muscle cell. So we have these infoldings of the plasma membrane here called T-tubules. And that allows that action potential to go deep inside the muscle cell. So here, these little structures here, these are organelles within the muscle cell. Remember, we're in the whole muscle cell here. So these structures here are organelles in the muscle cell. The name of these organelles is called Sarcoplasmic Reticulum. So I'm just going to put SR in these so that way you know all of this is Sarcoplasmic Reticulum. I'll spell it out for you. Sarcoplasmic Reticulum, Reticulum. Okay, again, anytime you see that prefix sarco, that means it's got something to do with a muscle cell. And this should sound a little familiar. It should sound like endoplasmic reticulum, and it kind of does. This is just fancy endoplasmic reticulum. So what makes it fancy? What's the deal? What's going on here? Well, sarcoplasmic reticulum stores, along with other things, stores calcium ions. So inside of this sarcoplasmic reticulum, you've got calcium ions just hanging around. They're just hanging out inside the sarcoplasmic reticulum, inside the organelle, inside the muscle cell. Now, also in that sarcoplasmic reticulum, you have these, oh my goodness, protein channels. And we're going to call those voltage-gated calcium ion channels. So when the action potential arrives down the T-tubule, it's going to cause the voltage-gated calcium ion channel to open. And what's calcium going to do? Calcium is going to go from high to low concentration, and it's going to leave the inside of the cell, or leave the inside of the sarcoplasmic reticulum and go to the, just hang out inside the cell. Now down here, we have another organelle. This is your myofibril. And that myofibril contains two proteins. It's made up of two proteins, actin and myosin. And you can probably see that Z line right here, or Z disc. And remember, that is one sarcomere, goes from Z disc to Z disc, right? So number one, that's one sarcomere. Okay, so when the action potential travels down the T-tubule, it causes the voltage-gated calcium ion channels on the sarcoplasmic reticulum to open. Calcium exits the sarcoplasmic reticulum, is now just inside the cytoplasm of the muscle cell. So now you've got all these little calcium ions floating around, and it is going to be around that actin, which is the big deal. Because if you remember what we talked about the last time, we said what caused the muscle contraction to start? What had to show up for that muscle contraction to initiate? Calcium ions had to show up. But I never told you where they came from. Well, they came from the inside of the sarcoplasmic reticulum. So now that we have calcium ions present in the cytoplasm of the muscle cell, we now know that we're fixing to get a muscle contraction out of that. So let's take a look at our bottom picture here. So this is a blow up of, like, this section right here. It's just kind of a blow up right there, or something like that. So you've got your myosin and your actin myofilaments. And remember, actin is made up of three structures. So you can't really see it very well, but here's one right here. That's these little rope-like structures. I think there's one right here too. It's kind of hard to see though. These little rope-like structures, these are called tropomyosin. And remember, their job is to cover the active site on the F-actin molecule, preventing myosin from binding. So that's why myosin is not forming a cross bridge with actin right now, because the tropomyosin is covering up those active sites so that the myosin can't bind. So these spherical structures, that's the F-actin part of the molecule, and that's what has the active site. That's where myosin binds, is to the active site on that F-actin. And then you should be able to see, it kind of looks a little different. Once you spot it, you should be able to be okay. This little three protein structure right here is your troponin complex. And remember, it's the glue that attaches. You got one of those that's called troponin T, that binds to the actin, that binds to the tropomyosin. You have the troponin I, which binds to the actin, and then you have the troponin C, which binds to the calcium. So that's why you have three parts, because you've got, again, it's the glue, holds everything together. So there's your other troponin complex right there. So right now, the actin, the F-actin molecule is, active sites are covered up by the tropomyosin, but here comes calcium. Got all this calcium now that has just come out of the sarcoplasmic reticulum and is going to be available to bind with the troponin molecule. And like we said before, when the troponin binds to calcium, that causes the troponin to change shape, moving the tropomyosin out of the way, exposing the active site on the F-actin. If you're going, what on earth are you talking about? Remember what we did the other day when we were talking about the actin molecule? This was page 2 of the actin molecule. We wrote it all down. So when calcium shows up, it binds to troponin C, causing troponin to change shape, moves the tropomyosin, exposing the active site on F-actin. This allows myosin to form a cross bridge, which results in a muscle contraction. We wrote that down last time, so I'm not writing it down here because we already wrote it down before. So let's watch the video so we can kind of see this in action. And hopefully it'll help make it a little bit more sense, hopefully.
And action potential introduced at the neuromuscular junction is propagated along the spinal level of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move to tropomyosin. When the tropomyosin moves, this uncovers the myosin binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
Okay, so that is pretty much the answer to your essay question. So this just kind of talks a little bit about what just happened. Here's another link that shows the same thing. So what happens next? As long as calcium is present, this process of myosin forming a cross bridge, myosin flexing, myosin releasing cross bridge, myosin going back to its original shape to rebind with actin, that's going to repeat over and over again. That's called cross bridge cycling. So I want to kind of show you the next step as now that the myosin is attached to the actin, what happens next?
Calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites to myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
And that just keeps going over and over again. Someone in one of the other classes asked me, and I just want to make sure you all know, these videos are in the PowerPoint, but they're also on the Moodle page. So I put right here like a snapshot of what that looks like on the Moodle page. So it's titled Video, Cross Bridge, and ATP Objective 4E. So I put them ABCD. I put the videos in order that we watched them in the class. So that's what the little ABCD part is for. So this is the 5th video that we've watched. So that's why it's called 4E. So anyway, and it's with Objective 4. So again, a lot of videos with Objective 4. But anyway, I wanted to make sure y'all knew that's what that was. These videos are on the Moodle page. So you don't have to go into the PowerPoint to watch them. Again, same thing, different video shows the exact same thing if you need to see it a different way. So again, that cross bridge cycling is myosin forms a cross bridge. You have the myosin head moving or flexing. Then you have myosin release with the ATP showing up. And then you have the myosin going back to its original position over and over and over again, as long as calcium is present. So this right here is the answer to your essay question. Again, I'm not going to ask you what step 1, 2, 3, 4. The next page 2 is also your essay question. So these 2 pages on your Moodle, on your PowerPoint, are the answers to that essay question. So you've got the, again, you put in your own words, the action potential that came from that motor neuron of the muscle cell, motor neuron is going to then get sent to the muscle cell, and it's going to move along the plasma membrane of that skeletal muscle cell. It's going to go down the T tubules, causing the voltage-gated calcium ion channels to open, releasing calcium from the sarcoplasmic reticulum. That calcium that came from the sarcoplasmic reticulum is going to bind to the troponin C of the troponin complex. Now, I told y'all, anytime you see the letter G actin, you can replace it with the letter F. It doesn't matter to me which one you call it. It's the same thing. G is singular, F is plural, doesn't matter. So the tropomyosin, once the troponin moves, it causes the tropomyosin to move and expose the active site on the F or G actin. Once the active sites on the F or G actin are exposed, that allows the myosin to form cross bridges. That's it. That's your answer to the essay question. So again, in your own words, and like I said, y'all, from what the ones I read first, I can't remember after, because then it was just a big mess of stuff. The essay questions looked pretty good. So you either got it right or you got it wrong. It was that kind of thing. Let's take a look at this page here. And this page here summarizes the entire process. So let's just kind of take a look at this and see what's going on. So we're going to start with number 1. So it is numbered. Okay, there's a number 1 right there. This cell right here is the axon terminal of the motor neuron. So you have your vesicles, you have your acetylcholine in the vesicles, okay? Your action potential is coming down the motor neuron. It doesn't have the voltage-gated calcium ion channels, but they're there. The calcium, when it floods in, is going to cause those vesicles to undergo exocytosis and put acetylcholine in the synapse. So you've got your synapse right here, the neuromuscular junction. So this whole thing here is the muscle cell. So you've got more muscle cell because you got a lot of stuff going on inside the muscle cell than you had before. So all of this right here, sorry, is the plasma membrane of the muscle cell. And remember, the plasma membrane of the muscle cell is also called the sarcolemma. So the action potential is is going down or going across the sarcolemma, and it's going to go into the T-tubule. So this is the action potential going into the T-tubule right here. That's number 2. You've got your sarcoplasmic reticulum right here. So this is your sarcoplasmic reticulum. It kind of looks kind of different, so I'm kind of hard to see it. So I'm kind of highlighting it here so you can tell what that organelle, where it's at. Hard to see it in a black and white picture, so that's why I'm kind of doing it this way. Okay, so all of this is your sarcoplasmic reticulum. Okay, and remember, its job is to store calcium ions. So it's storing those calcium ions in that sarcoplasmic reticulum. So when the action potential comes down the T-tubule, it's going to cause these, see that protein channel right there? It's going to cause that voltage gated calcium ion channel to open. And the calcium ions are going to get into the cytoplasm, cytosol of the cell. So it's going to leave the sarcoplasmic reticulum and enter into the cytoplasm of the cell. Now remember these voltage gated channels, the ions are moving from high to low concentration, so this is just passive transport. Remember that word? So they're just doing this through simple diffusion, okay? It's easy peasy, open the door, they flood out. So following what comes next, if you look down here, you've got your actin molecule and you have your myosin molecule. And notice we have cross bridge formation. And why is that? Well, because you got calcium ions floating around, that's why. So you got these little calcium ions floating around. And they're attached to the troponin. So what's happening here is what we talked about here. Calcium showed up, binds to the troponin C, causing it to change shape. We get cross bridge formation, and that's your muscle contraction. So that's what's happening in this step right here. So the calcium is going towards the troponin molecule. As we go around, we're following our circle here, clockwise around to the next step. Notice we do not have cross bridge formation. No cross bridge, which means no calcium ions. So what are the calcium ions doing? Calcium ions are going home. They're leaving. So because they're leaving, that means you don't have cross bridge anymore. Now we have got to put these calcium ions back where they came from. So this one's a little bit trickier. Again, they came out through passive transport. They came out that voltage gated calcium ion through passive transport. But in order to put them back, we've got to push them back because they're going into an area of high concentration. So this little protein channel right here, this is called the calcium ATPase pump, is what that's called. So it requires ATP to put them back into the sarcoplasmic reticulum. So we're going to just keep recycling the calcium. When we're not using it, it goes back into the sarcoplasmic reticulum, but we have to push it back using an ATP-powered pump. This is active transport, if y'all remember that. The ATP requirement means it's active transport. Okay, and that's pretty much the whole story on one sheet there. I know that was a lot. There's a lot going on in that picture, but it really summarizes what we've done so far very well, actually. I kind of like the picture. Y'all may not like it as much as I do, but really summarizes it. And here you can see that sarcoplasmic reticulum a little bit better because it's in blue, so it's hard to see it on the black and white page. It's a lot easier to see it here. So the steps of breakdown of ATP and cross-bridge movement, that's the next thing we're going to take a look at. So let's go back to our picture again. This is the last handout that you've got, which is this one right here. So we mentioned cross-bridge cycling. Cross-bridge cycling. So keep in mind where we're at in the process. Calcium ions are hanging around, triple myosin is moved, so the active sites are exposed. So that's the first thing that happens. So with step one, now y'all know I'm not going to ask you what happened with step one. I'm going to ask you what comes next. I'm not numbering these things, but we're just numbering them to keep them in the right order. So if you notice, we have cross-bridge formation. So after the active sites are exposed, we have cross-bridge formation. The next thing that happens, and I want y'all to notice this because I kind of typed it in as a different kind of thing. We have step number 2, which is called the power stroke. The power stroke, which is the myosin head flexing. So the myosin head is flexing. That is called the power stroke. And I do want you to be aware this is no ATP required for that to happen, which I know it sounds kind of weird because power, you think ATP. Well, that's not the case. The power stroke means no ATP. And then we go into step 3, which is now we have ATP show up. This means cross-bridge release. That's what the ATP molecule does. It allows the cross-bridge to break. No more cross-bridge because ATP showed up. Now, as we move around, the next thing that happens is the ATP that was attached split into ADP and phosphate. What's that called? ATP hydrolysis. So the ATP is split into ADP and phosphate. Now, it doesn't have a step 5 on this, but I'm going to give you a step 5 because, so we're going to have a step 5 here, which is called the recovery stroke. And this is the myosin head goes back to its original shape. So that's it was kind of part of step 4. It just has a little comma there. So that myosin head extends back to its original shape, ready to bind to myosin again. So we had it flexed over here, but we have to have it unflexed, and that's kind of what's happening at the last little piece there. It unflexes and is ready to bind to myosin again. And I do have on here the hydrolysis of ATP. I mean, I know we talked about it already. We've already drawn this out and everything, but here it is again, just in case. This is the hydrolysis of ATP. So you have your ATP molecule. Again, remember, hydrolysis means we had to use water to split that molecule. And that turned into ADP plus some inorganic phosphate plus some energy. And that energy got absorbed by the myosin head, allowing it to move. You don't have to know specifically how many kilocalories per mole it produced. That's okay. That was just part of the picture. Okay, so in your notes, that's what this is. So we've got the active sites were exposed. We've got cross-bridge formation that has occurred. The power stroke does not require ATP. It's just the myosin head flexing. The cross-bridge release, which does require ATP, and then the hydrolysis of ATP, which is the breakdown of ADP, which is the breakdown into ADP and inorganic phosphate. So that's that one. And then the last piece is the recovery stroke, and you can see that myosin head extending, ready to bind to actin again. Now, muscle relaxation is the active transport of calcium back to where it came from. So it's going through that calcium pump putting it back. So that's muscles relax during this phase. So you require ATP for the muscle to relax. So why is it that the muscles take twice as long to relax as they do to contract? If y'all have noticed, if y'all have ever gotten a muscle cramp at night, oh my gosh, it takes forever for that thing to not be crampy anymore. So why is that? Well, you got to have ATP so you can get the calcium back out of the cytoplasm and get it back in the storage facility in the sarcoplasmic reticulum. And that's active transport. It just takes a long time. To relax, it's just all passive transport, so it doesn't require as much time. So again, it's because of the movement of those calcium ions back into the sarcoplasmic reticulum through active transport. That's why it takes so long to relax. And this, I just pulled it off the internet. I just found a cut and paste thing. So if you need to kind of summarize it all up in your mind, that's not bad. Again, I just literally found it on the internet, and I thought, well, that kind of does it. So there we go. That was the very last slide. I told y'all I was going to keep y'all not too long today because I know y'all have a test in lab. So I wasn't going to keep y'all too terribly long.